Hi, this is Aryan, and this should be the fourth lecture of the Introductory Biology series. Okay, the lesson for this class is going to be on enzymes, but really we'll only be getting to those at the end. A lot of it is just build up to the details of how they work based on some chemistry and a little bit of physics. Alright, so bioenergetics is where I'll be starting for today. Basically, it's how we discuss the flow of energy in natural systems. So, for example, yesterday I woke up, ate some food, and then went for a run. You could consider the process of me eating food like inputting energy into my body, which I then used up during my run and other various activities throughout the day. Now, this is a word you might have heard before, metabolism, and it's really just any chemical reaction that your cells can use to make energy. And an excellent example of this is carbohydrate metabolism, which is basically when our bodies break down a carbohydrate and get energy from the process. An example of carbohydrate metabolism is when glucose, with the formula C6H12O6, can be broken down in combination with 6 molecules of oxygen gas to get 6 molecules of carbon dioxide, 6 molecules of water, and some energy that the cell can then use. Of course, this energy came from somewhere, and we can actually trace back that energy flow all the way to the sun. So the sun releases energy in two ways. One is heat, which you can feel if you go outside, and the other is light, which you can also experience by taking a look outside. Now, this light is used by producers, which are plants in a process called photosynthesis, to store energy in the form of sugars like glucose. And the process of photosynthesis is actually really cool and intricate, so if all goes to plan, I'll be making a lecture on that later. So currently, all of our energy is being stored and used by plants. Well, from this point, it can potentially go two ways. If the plants are eaten, the energy stored in their carbohydrates will be passed on to consumers, which are animals. And in the class of consumers, obviously there are some herbivores, which means they just eat the plants, and then there are carnivores, which then eat the herbivores that eat the plants, and so on. We could have carnivores that eat the carnivores that eat the herbivores, etc. Basically, all animals either directly or indirectly get their energy from plants. Now, both plants and animals might not be directly consumed and instead live a full life and die, which is where decomposers come in. Decomposers essentially take whatever useful nutrients they can from dead organisms, and they can be fungi, bacteria, and even some animals like worms. And in the end, the energy of the decomposers will eventually be used up just as heat. We can use bioenergetics diagrams like this to analyze where the energy from a food item, for example, came from. So let's say if I'm eating pizza, the ingredients might be tomato sauce, dough, and cheese. So dough and tomato sauce are both products from a plant, which got their energy from the sunlight. So that's part of the bioenergetics of the pizza. Now, cheese, on the other hand, is a modified byproduct of an animal, a cow, which got the nutrients and energy to make milk from a plant, which in turn used photosynthesis to get energy from the sun. So in the end, all of the energy in our diet can be traced back pretty much just to sunlight. Okay, now we're going to take a look at energy. Energy is technically defined as the ability to do work, but that's more of a physics definition that you probably don't really need to know right now. Something that would be pretty useful to remember, on the other hand, are these two types of energy. Kinetic energy is the energy of motion. So, for example, if we have this ball and it's rolling down the hill, it has to have energy because it is moving. And on the other hand, there is potential energy, which is the energy of relative position or structure. And that's kind of an abstract definition, but if we consider two more balls on this same hill, with one at the top and one on the bottom, the one on the top has the potential to have kinetic energy if it rolls down the hill, while the one at the bottom really doesn't have any. And that's what's meant by energy of relative position. As for the second part of the definition, we can think of chemical bonds as a different type of potential energy in the structure of the molecule. Basically, you can consider each bond in a molecule to have energy stored in it. Alright, if you're breaking a bond, you're probably going to be releasing energy. And whenever you release energy, the chemical reaction you get from that is called exergonic. For example, if I had gasoline and I set it on fire, which would be just breaking a ton of chemical bonds, and then I would release more fire. The reaction would be breaking the bonds, and then the heat from the fire is the energy released from the reaction as a byproduct, which means that this reaction is exergonic. Now, the opposite of an exergonic reaction is an endergonic reaction. In an endergonic reaction, energy is added. So one example of this is actually photosynthesis, in which sunlight, which is a form of energy, is used to make carbohydrates, specifically glucose. Okay, so I've been talking a lot about reactions, so I'm going to clarify some vocabulary. The reactants are the chemical substances present before the reaction, and the products are the substances present after the reaction. So for example, a common chemical reaction is O2 plus 2H2 can react to form H2O. And in this case, O2 and 2H2 would be the reactants, while water, H2O, is the product. Alright, now let's take a look at this quick practice problem. This is the chemical reaction I showed you 2 slides ago. C6H12O6, also known as glucose, plus 6O2 reacts to form 6 carbon dioxide and then some energy. Now I'll give you a few seconds to work on this problem, in which you can pause the video, get through it by yourself, and then I will go over the answer. Alright, now this question is actually pretty straightforward if we consider the definition of endergonic and exergonic. Endergonic means that the reaction uses energy, while exergonic means that it outputs energy. As you can see, energy is in the products side of the reaction and not the reactants side, so this is definitely exergonic. Even if I didn't include the energy, it should be still pretty clear that this is an exergonic reaction because a large molecule is being broken down into many smaller molecules, which means that the potential energy in chemical bonds are being released, which always is an exergonic reaction. Alright, so this is a graph of free energy levels over time for an endergonic reaction. Free energy basically means any energy in the system, and as you can see, it starts at this lower dashed line for the reactants, then increases and falls back down to a higher level for products. If we compare the free energy of the products, we can see that it's greater than the free energy for the reactants, because the red dashed line for products is higher on the energy axis for this graph. This means that the change in free energy, or delta E, is greater than 0, because that's just the final energy minus the starting energy, or the energy of products minus the energy of reactants, which must be greater than 0, because the products have more free energy than the reactants do, as you can see from this graph. So to summarize, for endergonic reactions, the change in free energy is positive or greater than 0. And this makes sense if we actually think about it, because endergonic reactions, by definition, add energy. So of course, there's an increase in free energy. Okay, this is the graph of energy levels over time for an exergonic reaction. This is pretty much the opposite of an endergonic reaction graphically, in terms of the placement of products and reactants' energy levels. The reactants' free energy is all the way up here with the products, while the products' free energy is far, far lower. So of course, this means that the change in free energy, or delta E, is less than 0, as the free energy of reactants is lower than the free energy of the products. So we are subtracting a big number from a smaller number, resulting in a negative number for the change in free energy. And of course, this makes sense because the definition of an exergonic reaction states that it releases energy. And as you can see from this graph, there's a loss of free energy overall. Also, in biology textbooks and classes, this quantity of free energy is called Gibbs free energy, and is generally denoted by a delta G instead of a delta E. That's just something to keep in mind. Now, there's another term we need to learn. Activation energy is the size of the hill in the free energy graph. Basically, it shows that a little energy is always needed to initially be inputted just to get the reaction going for both endergonic and exergonic reactions. Now, what enzymes do is they reduce this hill of activation energy significantly. This means that it's a lot easier for the cell to perform activities because it takes far less energy to get the reaction started. An excellent example of this is an enzyme called salivary amylase. Salivary amylase is found in your saliva, and it helps to break down complex carbohydrates. If you've ever taken a bite of bread or a saltine cracker and just held it in your mouth, you'll notice it at first gets soft and tastes savory, but over time it begins to taste sweeter. This is because the starch in it, which are long branching chains of sugars bound together, that are called polysaccharides is being broken down to individual monosaccharides like glucose by salivary amylase, which is pictured here. And while the starch chains taste savory, the individual glucose molecules taste sweet. Now, if we don't have salivary amylase, theoretically over the course of probably hours or more, the starch chains would eventually break down purely just by chance. But having salivary amylase reduces the activation energy so much that you can taste the difference in less than a minute. So that's just a cool example of how enzymes work, and pretty much every biological reaction in your body is going to have an enzyme involved. When we get to the process of DNA replication in a later lecture, for example, you'll see that there are a bunch of enzymes used at every step in that reaction, or rather series of reactions. Okay, now we're going to quickly review the laws of thermodynamics. These are not at all important to know for this class, but I'd like to review them just because they can be important if you ever end up studying chemistry or biochemistry in high school or college. The first law states that the total amount of energy in the universe is constant. And I touched on this a bit earlier, but basically no matter what, energy can neither be created nor destroyed. Instead, it's just converted to different forms. An example of this is when energy in the form of sunlight is converted to energy stored in the chemical bonds of a sugar molecule during photosynthesis. Okay, the second law of thermodynamics states that entropy or disorder will always increase. Basically, entropy is a measure of disorder in a system. So like all life, all life has pretty low entropy because all life is pretty well organized, while, dirt has higher entropy because it's not nearly as well organized. Now, if we consider outer space, that's probably an environment with the most entropy, because for the most part it consists of lone hydrogen atoms floating around in a near vacuum, which is of course not organized at all. Entropy also has a kind of inverse relationship with the amount of energy in a system. So more energy in a system usually means less entropy, while less energy in a system usually means more entropy. So let's consider a simpler example of entropy. My bedroom is kind of messy right now. I haven't made the bed. There are some books on the floor that I've just been leaving there for a while, and I have to organize, other stuff. If I put no energy into cleaning up my bedroom, it'll remain disorganized and maybe even get more disorganized. But if I take some time to clean everything up, then the room will be organized. So if I put energy into the system of my room, it'll be more organized and therefore lower entropy. But if I put no energy into it, it'll stay disorganized and have very high entropy. Biological systems like cells work in a similar way. Finally, the last law of thermodynamics states that as temperature approaches absolute zero, entropy approaches a constant. This really isn't important for biological systems. In fact, I don't even think the textbook I'm using to create these lectures even includes this when we're talking about thermodynamics. But basically, it means that you get colder and colder. That when you get colder and colder, there is less exchange of energy. So energy tends to stay where it is because of the increasing coldness, and this means that the entropy will change less and less as a system gets progressively colder. Again, this isn't super important, but it's useful to know, and if you ever end up taking chemistry, these laws will definitely come up again. Okay, so I've spent a lot of time talking about energy in really abstract terms, like the cell has a certain amount of energy, or energy is transferred, or glucose plus oxygen gives you carbon dioxide plus energy. But what does energy really mean in the context of cell and life? When a cell makes energy, it needs a way to store it so it can be retrieved and used whenever and wherever it's needed. And this is done through the molecule adenosine triphosphate, also known as ATP. ATP's structure is really interesting. On the right here, there is an adenine plus a ribose sugar, which is called adenosine, but that's not really important to remember. What is important in this section, is the section on the left here, which consists of 3 phosphate groups. Each of these phosphate groups has a negative charge, and when you put these 3 negative charges extremely close together, they tend to repel each other. But in this case, they're stuck together with covalent bonds. And the fact that these negatively charged phosphates are forced to be right next to each other, even though they really don't want to be, results in a ton of tension on the covalent bonds connecting them. Therefore, when one of these covalent bonds is broken, that tension is released in the form of a fair amount of energy. So we can say the reaction is ATP can be broken down into ADP, a phosphate, and some energy, where ADP stands for adenosine diphosphate and is really just adenosine triphosphate with a phosphate group missing. Okay, so if we recall a while ago, I said that glucose in combination with 6 oxygen gas molecules is broken down into 6 molecules of carbon dioxide and some energy. This is the main way animal cells get their energy, and the process is called cellular respiration. It's by no means straightforward, and it's not as if the energy from the bonds in the glucose is magically being converted to energy stored in ATP. But we can leave it like this for now. We'll go over the actual process in the next lecture. Okay, so how much energy does cell respiration actually make? Well, it turns out approximately 34 molecules of ATP worth, which is actually a fair amount. Alright, so we've learned about the energy of biological systems, how reactions work. Now let's get into the details of enzymes. And just to review, enzymes reduce the activation energy of a reaction, so they basically make that hill in the energy versus time graph a little bit easier to get over. Now it's time to go over some terminology. Substrates are reactants that bind to an enzyme, and an active site is the location where they bind. Basically, the substrate is just a fancy word for the molecule or molecules that the enzyme is helping react to get whatever the products of the reaction will be. Also, something important to keep in mind is that enzymes aren't immortal and will eventually degrade if left in a harmful environment. This could be a variety of things, from too hot to too cold or maybe too dry. If you ever end up in a biology lab that uses enzymes, then you'll definitely notice that they have a lot of refrigerators to store them in. This is because many enzymes will degrade much slower if they're left in the cold. So if you ever buy enzymes in bulk for some sort of experiment and you want all of them to last for years as opposed to a shorter period of time, refrigeration is the way to go. So let's take a look at an example of a theoretical reaction involving an enzyme. We have here this big enzyme in black and 2 reactants, the green square-shaped protein and the red triangular-shaped protein. And they bind to specific sites on the enzyme designed for them to fit perfectly into. Then energy is added, probably in the form of 1 or more molecules of ATP, and then the enzyme breaks that ATP down into ADP and a phosphate. And this energy from the ATP can then be used to change the structure of the enzyme such that the 2 substrates are pushed together in just the right way so that bonds are formed, and this results in a new molecule, or rather protein, I think, that is the square bonded to the triangle. And this newly fused protein will then go off to serve its purpose, leaving the enzyme ready to repeat this process all over again. Okay, here's another example. Here's another example of an enzymatic reaction. In this case, the substrate is a big yellow protein that binds to the enzyme and is then broken down into 2 smaller products, a pink oval and a red triangle that can then be used for something else in the cell. Also, you'll notice that energy was actually released in this reaction as a chemical bond was broken instead of formed. This means that this energy can be potentially used to make ATP that'll be used later, or maybe this enzyme can be used maybe the energy can later be used by the same enzyme in a different reaction. Alright, this is a bit more of a unique practice problem. I'm asking you, of the reactions I showed you on the last slide, which one is endergonic? The one where the 2 substrates are combined into 1 large product, or where the large substrate was broken down into 2 smaller products. As always, I'll give you a few seconds to work through this, in which you can pause, and then I will go over the answer. Okay, let's think about what exactly an endergonic reaction means, which is that energy is inputted, and the only reaction in which energy is inputted instead of gained in this case was A. Because remember, ATP had to be used in order to make the two substrates bond together. B, on the other hand, energy was released. Okay, next we're going to go over enzyme inhibition, and this is when a cell wants to temporarily stop an enzyme from working. For example, if an animal cell is at a point where it has enough energy, it may want to relegate some glucose to storage instead of just breaking it down. The way this could be done is by producing enzyme inhibitors that stop enzymes from breaking down glucose so that it's saved for later. Enzyme inhibition can be done in one of two ways. The first being competitive inhibition, which is where the inhibitor protein binds in the active site, which is where the substrate would normally bind. This inhibitor will stay bound much longer because it's designed to just kind of fill that space and isn't actually modified, so there's no reaction occurring. And this is called competitive inhibition because the inhibitor effectively outcompetes the substrate for use of the active site. The other form of inhibition is called allosteric inhibition. In this case, the inhibitor binds to a location other than the active site, and this binding results in the enzyme changing shape so that the active site is no longer available or changes structures in such a way that the substrate cannot bind. Okay, now that we understand how enzymes work, let's take a look at an example. First of all, enzymes are almost always named after their substrate or substrates, but with the ending of "ase" instead of whatever ending the substrate originally had. So for example, we'll be taking a look at the enzyme that degrades lactose, which according to the naming scheme is lactase. However, before looking at lactase, we should first understand lactose, which is Latin for milk sugar, because it's usually found in milk and it's actually a disaccharide, meaning that it's a carbohydrate made of two monosaccharides, specifically glucose and galactose. As you can see, this is the reaction. Glucose and galactose are combined to form lactose and a molecule of water. The red boxes indicate the parts of the monosaccharides that actually form the covalent bonds with each other, but that's not really important. Anyway, lactase essentially reverses this reaction, breaking up lactose into two-component monosaccharides. Lactose intolerance is a pretty common condition in which an individual has a lactase deficiency. So basically, their lactase enzyme isn't produced correctly and therefore cannot actually break down lactose. Okay, and to give you a sense of the complexity of enzymes, this is what lactase looks like. And it's pretty crazy considering the relative simplicity of its substrate as basically just two sugars bound together. All right, I'm going to be wrapping up this lecture with a list of vocabulary to remember. Metabolism consists of any chemical reaction used by the cell to make energy. Kinetic energy is the energy that is the energy of an object in motion. Potential energy is the energy of an object that's in a certain relative position or resulting from structural. Endergonic reactions are reactions where the energy is inputted, while exergonic reactions, the energy is released. Reactants are the substances used in a reaction, while products are the results of a reaction. And adenosine triphosphate, or ATP, is a molecule used to store energy. Substrates are the reactants that bind to an enzyme, and the active site is the location on an enzyme that the substrate actually binds to. And I believe that about wraps it up for the fourth lecture in our introductory biology series. I'll see you all next time.